When sampling in the field, you'll need a number of supplies. So you'll need some resealable containers that are airtight. You can use plastic bags, or you can use a plastic container that has a seal inside the top. That's for desiccating the samples after they've been collected. You'll need packets to collect the samples in. These are number 7 size coin envelopes. You'll need a label affixed to them. They could be pre-labeled, or you could have labels that you put on. They should have a unique identifier, and it should be printed, as well as a barcode for it for scanning. You can see these are taped on. That's the most rudimentary approach, but you can also print them on sticky labels and then peel and stick them. You'll need some fasteners to make sure that once the sample is collected that it's secured. That could be a paper clip or one of these clips. You'll need scissors for cutting the leaves for your sample. You'll need isopropyl alcohol, 70%. Get it from a pharmacy for cleaning, and you can put that in any container. You can buy labware, but clearly here this is just a reused bottle for contact solution, eye contacts, and then some sort of alcohol wipes or paper towel for cleaning the scissors. So when you go to sample, you'll take your sample packet, and it's just one sample packet per variety. So you'll want to talk with the respondent about where that variety is located. In this case, we can say, oh, this is a patch of rice. Clearly this is grass, but it has the same sort of leaf structure as rice. And you would then look at the boundaries. You probably GPS the boundaries first, just to be familiar with them. Then you'll want to walk into the plot and start collecting plants while walking in a V pattern. Now, you want to collect a certain number of plants. In the case of rice, as a inbred selfing pure line type of variety, we'll want to be collecting 20 plants, leaves tissue from 20 plants. And so you'll pick a first plant, and you'll pick a healthy leaf that is new and growing, and you will clip a 4 centimeter portion, or whatever the protocol indicates in terms of size of amount of leaf to collect, and put it in the sample pack. And then you'll walk 10 to 20 feet in order to sample the next one. And again, following a Z pattern, so that you will sample different parts of the plot on the interior. If the plot's really large, you of course won't walk the whole plot, but just a portion of it. But of course, being in the interior, this is rather small area, so we will not walk that much. And again, you want to collect the same amount per plant, a healthy new leaf. And so after you've collected your 20 plants, you will seal up this packet. As required, you might take GPS points at each plant that you sample. So you would want your device then that you're taking GPS points with to be available to you, and when you take your sample, to also then capture the GPS point that corresponds to that particular sample with your device. So in this case, a phone. So once you've got your sample, you'll take that back with you, and you will seal up the bag. You can dog ear the corners to help it be sealed, and clip it, and then put it in your resealable container that has desiccant in it. The desiccant will draw the moisture out. In this case, it's blue silica indicating gel. And you will squeeze out the air and seal it. And this then will go in your backpack. It's important that this be kept in a coolish, dry place, and so a light colored backpack is great for that. In the field, it won't take on too much heat, and you'll just want to make sure not to store it in the trunk of your car or leave it in the car unattended for long periods of time. At this point, there will be some contamination, of course, on your cutting device, so you'll want to clean that with the alcohol. So you'll clean the the blade by washing it with alcohol, and then of course cleaning it with the wipe so that all the residue is removed and you don't have contamination from variety to variety. The particular label on the packet will be scanned into the data application so that it is associated with information that's been collected about the plot of this variety, and that completes the sampling protocol.